Hi, I'm Daniel.
I'm Brandon.
Today we will be presenting over our research on the release of active pharmaceuticals using capped hyperbranched polyesters.
So first we want to introduce some of the problems with conventional drug delivery at the moment. And if you look at this graphic, you can see as you first consume your drug, it instantly spikes in your blood concentration. And as it's absorbed, it falls exponentially in your blood concentration, leading to potential toxicity when you're above the effective range and ineffectiveness when you're below the effective range. Also, you can see it spends very short periods of time in the effective range. Theoretically, with a sustained release application, you can release your pharmaceutical over a long period of time at a constant rate.
So our platform for sustained release was hyperbranched polyesters, and what we would do is we would covalently bond or encapsulate these active pharmaceuticals to the hyperbranched polyesters. The advantages of hyperbranched polyesters over other methods of sustained release is that hyperbranched polyesters are non-toxic, specifically the monomers that we had on our hyperbranched polyesters, and they were inexpensive and biodegradable. They're also actually on the generally regarded as safe list. And another advantage of hyperbranched polyesters is you can adjust the monomers that make up the hyperbranched polyesters and have a manipulation of release rate. So the basis of a hyperbranched polyester is an ester bond, and an ester bond is formed when a carboxyl group and hydroxyl group bond together, and you have an ester bond with water as a byproduct. If you want to form a linear polyester, you do two difunctional group monomers, bond them together. You have two ester bonds, and again, H2O is a byproduct. Our end goal was hyperbranched polyesters. And hyperbranched polyesters require a monomer that at least has 3 functional groups and a monomer that at least has 2 functional groups. And this obviously forms a very highly branched pattern with many groups on the end that you can cap pharmaceuticals to. We made 3 hyperbranched polyesters: a glycerol succinic acid hyperbranched polyester, a glycerol adipic acid hyperbranched polyester, and a trimethylpropane or TMP adipic acid hyperbranched polyester. One important thing when synthesizing these hyperbranched polyesters was the ratio of moles of functional groups. And using Flory's theory, which is very dependent on the number of functional groups on each monomers, for a system with a monomer that has 3 functional groups and a monomer that has 2 functional groups, we determine the ratio of moles of functional groups must be greater than 2 or less than 1/2. And choosing 2 or 1/2 depends on what end groups you want for your hyperbranched polyester. If you want a hyperbranched polyester with hydroxyl end groups, you'd put the monomer with hydroxyl groups in excess. If you want a hyperbranched polyester with carboxyl groups, you put the monomer with carboxyl groups in excess. And so the small molecule therapeutics or active pharmaceuticals we decided to test for our research was salicylic acid, which is the active metabolite of aspirin. It's actually aspirin in the body. Aspirin just has an acetyl group to reduce irritation when it enters the body. We also use naproxen, a common non-steroidal anti-inflammatory drug used for pain and inflammation. And we use hydrocortisone, which is a little different because it's usually topical and used for redness and swelling on the skin. As you can see on the diagrams, salicylic acid and naproxen both have carboxyl groups, but hydrocortisone has hydroxyl groups. So this is what requires the different end groups on the hyperbranched polyesters. And so when capping these small molecule therapeutics to the hyperbranched polyesters, we use the equimolar ratio of carboxyl to hydroxyl because we thought this would be the most efficient. And here's an example of capping of salicylic acid to a glycerol adipic acid hyperbranched polyester. As you can see, the hyperbranched polyester's hydroxyl group and the salicylic acid with its carboxyl groups, these two are placed in a flask with a little bit of catalyst, such as toluene sulfonic acid. And we place this in an oil bath of around 140 degrees Celsius to 150 degrees Celsius to achieve a capped hyperbranched polyester.
So during and after synthesis, we would have to characterize our polymer to make sure we were making what we wanted. And for this, we used what's known as size exclusion chromatography, or SEC. And so how SEC works is you would dissolve your polymer inside of a solvent, in our case THF. And as you flow your solvent through your column, the molecules of different sizes will spend different amount of times in those porous gels. And so as you can see, the bigger molecules will spend less time inside of these gels, leading to them being eluted earlier. And then the smaller molecules will spend even more time, leading them to be eluted later. What we used this for was to check what stage of our synthesis we were in. And after capping, we would see an increase in molecular weight by comparing both of the SEC data. We also used NMR spectroscopy in order to analyze the exact composition of our polymers. And so what we would do is we would double check the ratios of our monomers to see if they match what we wanted theoretically. And we would also analyze to determine what the exact composition was in terms of pharmaceutical. And in our cases, we noticed that we loaded around 30% on average to our hyperbranched polyester. After we characterized our polymers, we would move on to what's known as an in vitro study, which is basically replicating a living organism outside of the body in a place such as a lab. And what we did in our case was we used rat liver microsomes, which facilitated the enzymatic hydrolysis of our hyperbranched polyesters. And as you can see in this graphic, in enzymatic hydrolysis, the substrate bonds to your enzyme, and that enzyme facilitates the cleavage of those bonds. In our exact experiment, we would load a vial with a buffer, pH 7.4, our capped hyperbranched polyesters, and then we would inject the enzymes in, taking samples at different times in order to analyze the release kinetics.
So our method for analyzing these samples was high-performance liquid chromatography, or HPLC. In HPLC, its separation is based on polarity. So we have a water acetonitrile gradient. At the beginning, it's 90% water to 10% acetonitrile, and it slowly transitions to, by the end, it's 100% acetonitrile to 0% water. So when the different molecules in these samples have a polarity similar to the gradient, they will dissolve in the sample in a loop, and a UV detector will detect that, and a retention time will appear. And we can use this retention time to identify when pharmaceuticals are actually showing up on our samples by creating a standard, such as the one shown above, and then relating that standard to the actual samples to identify where the pharmaceutical actually is released. And then by using the peak areas, there's different peak areas as the time went on, we could determine the amount of pharmaceutical that was actually released through calibration of graphs.
So like Daniel said, we had to use calibration graphs in order to relate the arbitrary area counts from the HPLC to a known concentration. And what we used is we shot up samples of 0.1, 0.2, and 0.5 milligrams per milliliter. And what then we did is we modeled that next to the area counts from our HPLC data in order to create this equation that allowed us to further analyze the release kinetics. And so our initial results, we saw right away when we were trying to hydrolyze the hyperbranched polyesters. Without enzymes, they would take very long periods of time, as shown here, to hydrolyze even to a significant degree. And this showed that, in comparison to the hydrolysis with enzymes, this showed that our polymer had a stable shelf life and only degraded significantly in the presence of enzymes. Besides that, we noticed, Daniel mentioned that we made two polymers with different trifunctional alcohols, TMP and glycerol. We noticed right away that the TMP hyperbranched polyester wouldn't degrade from SEC data. So that allowed us to continue with glycerol as our trifunctional alcohol of choice. In terms of the actual release of the pharmaceutical, we noticed with all of our trials, we got a linear release rate that was replicatable between trials, which indicated that zero-order kinetics, which is extremely desirable in the pharmaceutical industry. Besides that, we noticed that we were successful in manipulating the release rates of each of the pharmaceuticals by alternating between monomers. And if you look at this graph on the right, or on the left, you can see that one of these, the red, is a polymer made out of glycerol and adipic acid, compared to the blue, which is a polymer made of glycerol and succinic acid. And you can see there's a significant difference in the percentage released and the release rates.
So our conclusion from these results, first of all, as Brandon mentioned, TMP did not hydrolyze, the TMP hyperbranched polyester did not hydrolyze where the glycerol hyperbranched polyester did. And this is very interesting as both have 3 hydroxyl groups and very similar in structure. And we deduced that this is likely due to enzyme selectivity, as enzymes require a very specific structure from the substrates that are being put in order for it to function, and it's likely that the TMP hyperbranched polyester did not fit that criteria. We also, as Brandon noted, we also noticed a linear release rate, and we think this is actually zero-order kinetics, and this was proven when we tested an in vitro study with only 10% of the original amount of hyperbranched polyester, as we still achieved a similar release rate. And this could be, we hypothesize, for 2 reasons. First of all, there's a thing called Michaelis-Menten kinetics, which states when the substrate concentration is far greater than the enzyme concentration, the substrate concentration doesn't matter, which is what is important for zero-order kinetics. And instead, the rate is dependent on the concentration of the enzyme. So for the first part, for our 2 hours, we were able to achieve a linear release rate in zero-order kinetics. Another hypothesis is the fact that when hyperbranched polyesters are placed in the buffer, instead of a solution, and actually an emulsion is formed. So it's not actually just it's not a dissolved hyperbranched polyester, it's particles. And so when the particles are in the buffer, they must be eroded by the enzymes going from the outside in, and this creates a linear release rate. And then the final result was the time release, showing that succinic acid has a slower release rate than the glycerol adipic acid capped hyperbranched polyester. And we again think this is due to the fact that when the buffer and hyperbranched polyester are placed together and stirred, the hyperbranched polyester actually forms particles and it's actually not dissolved. And because succinic acid is a smaller molecule, it creates a more rigid hyperbranched polyester, which creates bigger particle sizes. So it takes longer for the enzyme to go from the outside to the in of the particle, and therefore it creates a different release rate. So for future plans, we like to look more into enzyme selectivity, as we think this plays a big role in a lot of different areas of our research, as we also noticed that naproxen actually released slower than salicylic acid every time. We also like to test other enzymatic hosts. Earlier in our process, we had tested with blood and rat S9, but we had not finalized our analyzation process. It just happened we were using when we found out about HPLC, we were using rat microsomes, so we decided to stick with that. We were pretty sure that blood and rat S9 will also work as enzymatic hosts. And lastly, and most importantly, we found that hyperbranched polyesters are a viable method of sustained release. And we're working with Michigan State University to do in vivo studies with rats. And possibly in the far future, hyperbranched polyesters can be used for veterinary purposes and even third world countries because of the inexpensive materials that make up our hyperbranched polyesters. And here are our references. And we'd specifically like to acknowledge our mentor, Dr. Patrick Smith, for always encouraging us and helping us through this whole process.